All right, well, I'd like to welcome everyone to today's edition of the Polyploid Webinar. Today we have two great speakers, and up first is Will Booker from Florida State University. Will is currently a PhD candidate in the Department of Biological Science at Florida State. He is broadly interested in speciation and the process of diversification, particularly as it relates to gene duplicate evolution. His work currently involves investigating the evolution of populations in the polyploid gray tree frog complex and the evolution of the insular Bothrops Jararaca snake complex. With that, I'll let Will take it away.
Thanks, Mike. Yeah, so today I'm going to be giving a talk on the complex history of genome duplication and hybridization in North American gray tree frogs. So just to kind of introduce the system, gray tree frogs are composed of two currently recognized species. There's a diploid Hylochrysoceles and a tetraploid Hyloversicolor. They look identical. The only reliable way to distinguish them outside of karyotyping is their call. And so Hylochrysoceles has a fast pulse rate call, whereas Hyloversicolor, the tetraploid, has a slow pulse rate call. In actual experimental tests creating synthetic polyploids showed that this is a direct effect of the larger cell size in polyploids. So they can make synthetic triploids, and the call decreases in pulse rate in the direction of the tetraploid. And so it's a unique case of like automatically evolving at least some reproductive isolation whenever these polyploids formed. The complex pretty much exists all over Eastern North America and up into Canada, from central Texas all the way to the East Coast. They are separated by range somewhat, with only the tetraploid in the northeast above Pennsylvania, and the Chrysoceles only really being the only one in the southeast. But there's a broad overlap of sympatric zones, sort of in different parts of their range, and then you find pockets where you find only tetraploids and only diploids in certain places. And so it's, exactly what's causing that is kind of unclear, but you do find some mosaics in those areas. And so previous work up until what I'm going to be talking about today to try and describe the diversity in this system, the last comprehensive study of the evolutionary history of this group was conducted by Holloway et al. In 2006. And they sequenced a couple of nuclear and mitochondrial genes, as well as looked at their calls, and came up with this hypothesis that the tetraploid Hyloversicolor arose independently multiple times, including with the existence of extinct ancestors contributing to all three of them, and the extinct ancestors only creating one of the lineages. And then currently, the tetraploid complex is all sort of hybridizing, even though they arose independently. And so, but there's been some critiques of this. Jim Bogart and B 2013 discussed sort of that allopolyploid is not the only way to explain the data. There are scenarios of autopolyploidy that are justified with the data as well. And Holloway et al. Study, even though transformative at its time, was limited by just a few loci. And then also previous work suggested autopolyploidy with a little bit of mixed chromosomal inheritance suggested possibly for in the process of diploidization. And so it could also be kind of a segmental allopolyploid or a autopolyploid that's undergoing the process of diploidization. And so the present work was really to try and reinvestigate this complex with the data that were available when we started this project in 2016. And so we decided to use anchored hybrid enrichment and at the FSU Center for Anchored Phylogenomics. And we started with 127 individuals from pretty much throughout its range, And we tried to include all the samples from Margaret Tatchek's paper in 1994, which looked at the mitochondrial evolutionary history of the group, as well as Holloway et al. 2006. And then we added some from new samples from collections to try and fill in some gaps. And so we had a little over 247 single copy loci, a few 100,000 nucleotides, some variable sites, and little missing data. And so I'm not going to go into the first part about this, of sort of what we studied, what we found, but previous work suggested multiple independent origins. And if you look at the full mitochondrial phylogeny, you do see that there are independent clades of mitochondrial groups that are broadly geographically co-located in certain areas. And due to their affinity, especially with a diploid relative of the group, does kind of seem to suggest that there have been multiple independent origins, possibly between 2 and 3 independent origins. And then some nuclear data, if we're just looking at phylogenies, which I'll show on the next slide, kind of suggest this as well. However, the two species, again, are indistinguishable phenotypically, and the calls differ, but in a predictable fashion, just by an increase in cell size. And there's evidence of inter and intraspecific migration, which might create the same sort of pattern that we see, even if hyloversicolor did arise by autopolyploidy. And what I mean by some of the nuclear genes is we sort of try to use a method where we separated the subgenomes or the putative subgenomes in hyloversicolor to try and see their phylogenetic relationship to extant Hylochrysophilus by sort of separating the max divergent subgenome and the minimum divergent subgenome from Hylochrysophilus. And we do get a pattern where in the max divergent subgenome, we get high phylogenetic support for a separate clade outside of Hylochrysophilus. And then with the minimum subgenome, it does sort of separate into its own clade, but there's basically no support for that. And it kind of suggests the pattern we would expect under a segmental allopolyploid, where we are able to distinguish the max subgenome kind of, but the min subgenome, there's been some recombination that kind of makes, starts to pull it away from the diploid, but there's really no support for that. But when we looked at sort of our separation of min and max alleles, it's hard to say that that's justified. So on the blue dots there in the bottom, we have the pairwise genetic distance from the average Hylochrysophilus individual. But the min and max subgenomes of hyloversicolor are not that divergent and nowhere near the level of divergence of their most closest relative, Hyloaboboca. So it doesn't really support allopolyploidy in that sense and being able to separate the subgenomes unless it was a really, really recent ancestor or a relative of Hylochrysophilus. And so we wanted to look at this with different methods, and those methods were really focused on trying to separate pattern from process in this group. And so what I mean by that is, given a phylogeny that looks like this on the left, it can be explained by several different processes. So strict allopolyploidy could certainly explain this phylogeny, but also autopolyploidy with migration might explain this phylogeny for a given locus. And then as well, extinct lineage autopolyploidy and migration could explain this as well. So trying to sort of tie in whether or not there was an extinct lineage into these analyses. And so going forward, the goals of this next step was to try and really identify which lineages each hyloversicolor lineage descended from, test support for migration between lineages and species, estimate probability of polyploid speciation models, and estimate timing of events from the posterior from those other model tests. So the first thing we did is we tested some models of lineage, descendance, and migration using the program Migrate, developed by Peter Beerley. And so what we did is we selected 5 individuals from each mitochondrial lineage in both diploids and tetraploids, and tried to capture the nuclear genetic diversity that characterized that lineage. We randomly selected 50 loci, and we conducted 2 series of model tests. One test per polyploid lineage, testing migration and descendance patterns for the chosen lineage from each of the remaining polyploid and diploid lineages. And then one final test where we took that information to sort of test this broad model of evolution with descendance and migration. And so the results of that are a bunch of different tests that might be a little bit messy to look at. But importantly, what we found is that the northeast, the far northeast around Pennsylvania up to Maine area, that mitochondrial lineage was really the only lineage that had any support from being descended from Hylochrysophilus. And the other 2 lineages had very high support of being descended from that original polyploid lineage, sort of suggesting that there was a single whole genome duplication event or a polyploidization event, and that these other mitochondrial lineages arose via backcrossing with some other Hylochrysophilus population, introgressing their mitochondria and then expanding out further. And when we tested a series of different models where we tested some differently well-supported models, as well as some past hypotheses, that is the model that was best supported. We found most support for a single whole genome duplication event model, and the highest support for this stepping stone model, where Eastern Hylochrysophilus gave rise to Northeast Hyloversicolor, which then backcrossed and made Midwest Hyloversicolor, which then backcrossed with another Hylochrysophilus population to make the Southwest Hyloversicolor lineage. And importantly as well, nearly every model has higher support with migration. So migration from both diploids to tetraploids, as well as between tetraploid populations. The only one that didn't was migration from the Northeast Hyloversicolor into Midwest Hyloversicolor. And so part of that is just kind of saying that regardless of ploidy, there appears to be a lot of hybridization going on, whereas any population, any lineage is going to hybridize with whatever lineages are around it, regardless of ploidy. And we also see some support of that when we look at the genetic PCA axes. So that green squares there is the Northeast Hyloversicolor lineage. And we see that it's pretty far separated from all of the other Hylochrysophilus and Hyloversicolor groups, but most similar to that Eastern Hylochrysophilus in purple. And then whereas the other tetraploid lineages are sort of mixed in with this sort of central Southwest Hylochrysophilus group, which would be a pattern we would expect from backcrossing and hybridization to form those lineages. So the next thing we did was to actually test some models of polyploid speciation. And so we wanted to test specific polyploid speciation models using approximate Bayesian computation. This is a modified protocol or analysis from Roux and Pinnell 2015. And based on our previous results, we used Northeast Hyloversicolor and Eastern Hylochrysophilus populations. Now, if we used all of the models available to us, that gives us 45 possible models using identical priors to test with. So we needed to narrow this down to a smaller set of hypotheses. And so one way we did this is we looked at the population of allele frequencies to find evidence of disomy, tetrasomy, or heterosomy. So we would expect an abundance of alleles at 50% frequency in the population if it was heterosomic or tetrasomic, but the observed data doesn't show this. The observed data looks identical to our tetrasomic simulated allele frequencies, suggesting at least the bulk of inheritance is tetrasomic in hyloversicolor. Okay, so we came up with these 6 models to test from. Autopolyploid tetrasomic or allopolyploid tetrasomic with different patterns of migration. So either migration and no migration, migration in both directions. So from diploids to tetraploids and tetraploids to diploids, or just single direction migration from diploids into tetraploids. So we used custom scripts to generate the priors and calculate the summary statistics. Sorry. MS and SAM to simulate the sequences, and then the ABC-R package to estimate model probability in posterior. And so what we see is pretty much all of the support is between these 2 models of unidirectional migration from diploids into tetraploids. And the better supported model is an autopolyploidization model with unidirectional migration, and again, tetrasomy. And if we look at the estimated whole genome duplication timing from the posterior, we see that for the autopolyploid model, this places that timing around 100,000 years ago, which is what we see from the coalescent timing estimates from our mitochondrial tree, as well as what has kind of been hypothesized in the past that it was sometime after the Illinoisan glacial period. But whereas allopolyploid, for that model really to be the best model, it would have to have happened about 3 million years ago. And so that doesn't really, that actually is before the whole complex arose, and even before the evolution of the relative sister species. So it seems kind of unlikely, even though that model is somewhat high supported. And part of the reason why it's kind of hard to distinguish those 2 when we looked at linear discriminant analyses, basically when you start to add migration into these simulated models, autopolyploidy and allopolyploidy really start to look quite similar, which makes sense. You know, it becomes harder and harder to distinguish the different subgenomes and the different patterns when you start adding in migration into there. But really based on everything, based on the evidence of tetrasomy, as well as the evidence from the migrate test, as well as the ABC analysis, seems to all sort of suggest a single autopolyploid origin of hyloversicolor, with reticulation with hylochrysoceles forming the other lineages. There's lots of unidirectional migration from diploids to tetraploids, and high migration within species as well between different populations and lineages. And so on the right here is sort of the model of network model of evolution of this group from different extinct lineages of diploids, creating the extant hyloversicolor complex here. It's a lot to take in, so don't worry about not being able to see it right now. But it's really complicated. That's kind of the point of that right there. And so going back to sort of to the complex as a whole, to try and pull this into maybe some phenotypic aspects to look at. Remember that the diploid has a much faster pulse rate call than the tetraploid, and that's due at least mostly by the increase in cell size from ploidy. And so other work that I've been doing has been trying to look at the evolution of acoustic signaling in this group and sort of tie it to biogeography as well. And I'm just going to introduce a little bit of that here. But if we look at the diploid hylochrysoceles, we see there's lots of variation in calls throughout its range, from faster pulse rates with high pulse number, faster pulse rate calls with a very low pulse number, or a slower pulse rate calls with a very high pulse number like those in the east and those blue circles there. So there's lots of variation. So it kind of, this variation might give us some insight into the variation we might find in hyloversicolor. And so interestingly, what we see is in this southwest hyloversicolor lineage, which is the most recently formed lineage of all of them, and it's from, at least the evidence seems to support back crossing from the Midwest lineage with Southwest Hylochrysoceles. Those have a distinct call as well. And it's a faster pulse rate call in the direction of local diploids that's associated with that Southwest mitochondrial introgression event. But interestingly, when we try and make estimates of the energetic cost of these calls, this call is more energetically costly, and also by going in the same direction as local Hylochrysoceles, it reduces any sort of reproductive isolation that they may have, which would lead to more hybridizations, which we know is really costly due to the different numbers of chromosomes between the two species. Even though they do hybridize a lot, it's still very costly. And so it seems disadvantageous, and it's not really clear why that is. Maybe in a talk at some future point, I'll have more information on that, but that's sort of kind of an area that would be interesting to go into next. And with that, that's my talk. Thank you all for joining me. The picture there is the picture of the labs of Emily and Alan Lemon, consisting of these people on the right here. Like to thank some people who gave me input along the way: Carl Gerhardt, Jim Bogart, Dave Hillis, Joseph Travis, Camille Roux, Thibault Leroy, Travis Leduc, and Peter Beerley. This work, along with other work that's going on right now, has been supported by NSF, some internal grants from Florida State University, ASN, the Explorers Club, SSE, as well as SSB and Society for the Study of Amphibians and Reptiles. And that's everything I have.
All right. Well, thank you, Will, for a wonderful talk. As usual, we have plenty of time for questions here before the next speaker. So if folks have questions, go ahead and type them in the chat box. And it's always great if we can also then go ahead and ask those, turning on your video and audio. And there's Jonathan. Jonathan.
Yeah, so chromosome pairing, it seems to be mostly tetrasomic inheritance in this group. There has been some studies in the 70s between that Jim Bogart did that found some loci appear to be inherited disomically, but it seems to be, at least from our estimations, that it's mostly tetrasomic. The extent to which that varies in different locations, populations, it's hard to kind of know right now. All the lineages as a whole seem to be largely tetrasomic, but there might be some local differences and things like that. Let's see, next talk.
Right. Next question there is from Doug Zoltis. If Doug wants to turn on too, that's always,
okay.
So I kind of messed up my comment there. It was great stuff, but you mentioned subgenomes in the tetraploid. So if it's a true auto tetraploid, there's only one genome, not two, and
Right.
that tetrasomic inheritance. So I guess what do you mean by subgenomes in an autoploid?
Well, I refer to subgenomes as sort of before we really knew auto or allopolyploid, to try and distinguish between if it was an allopolyploid, which parts came from Hylochrysoceles and which parts came from another species and things like that. But yeah, I mean, in the tetraploid, at least as we see it now, there's not really subgenomes. It's all one thing.
Thanks. Great.
Oh.
Yeah.
I was going to insert a question here real quick. So do you think the genome structure would, if you got a whole genome sequence, I guess is what I was trying to ask, you'd be able to see larger blocks, because it seems like this analysis, you know, is based on the not having that linkage information. Do you think that would help reveal a little bit more about what's happening with this sort of auto-polyploidy these integration blocks to help date that? I'm just sort of curious.
Yeah, so I think there are
genomics in this group. Yeah.
So I think, you know, these were this is all Illumina data, so not very long loci. Longer reads would definitely help. I think particularly trying to look at regions around genes that have organelle targeted genes might be useful in trying to sort of see the kind of prove some of that stuff. And then trying to find some genes that maybe existed in some of those or were descended from some of those extinct mitochondrial hylochrysostomus lineages, to maybe try and piece together the biogeography of those genes and how they've been sort of passed down through different hybridization events and things like that. I'm not really sure how to sort of prove that, if we found that, how to sort of find that gene, you know, or sets of genes that look like that. But it would be really interesting if we could find those, because we're definitely losing some information with the mitochondrial genome phylogeny, because some populations, some lineages of hylochrysostomus that would be informative are currently extinct. So
cool.
Yeah, long read sequencing would be really great to try and parse some of that out.
Rob has a question there. Rob Denton. If Rob wants to turn on his audio and video, feel free.
Yeah, if you want to talk, Rob, or I can just answer the question as it is. So as far as climate and environment goes, I'm not entirely sure. I mean, I think climate, and it's particularly as it plays a role in the oscillations and glaciation cycles, I think was important, at least for maybe getting some lineages contracted into certain ranges and maybe driving lineage or species interactions, and also maybe reducing population sizes so that something like a mitochondrial introgression event might be more common, or more likely to fix in a population. But as far as climate and environment goes, like, I don't really know. As it relates to that, it might be interesting because it might, you know, because hylochrysostomus and hyloversicolor exist in this wide range of environments and climates, they might have been able to persist in more areas and have larger ranges through glaciation cycles, whereas other species might not. One species to look at in that as sort of a comparison would be Pseudacris crucifer, who largely have the same range as hyloversicolor and chrysostomus. And at least in some preliminary work I've done, seems to have shared some glacial refugia that they did too. But exactly why they exist in that wide range of climates is, I'm not entirely sure. And why certain lineages are some places and other ones aren't. If there is a reason other than just they happen to be there and be the one that fixed or outcompeted, I'm not really sure. Okay, let's see. From panelist Soltis. Do you think that diploids are truly extinct or just unsampled? So I don't think they're truly extinct. That mitochondrial lineage I think is truly extinct. As far as nuclearly, what currently exists, there's probably a lot that is in current lineages. But I do think that they're, at least in the Northeast region, where the original whole genome duplication event was, that mitochondrial lineage, I think, is definitely extinct, and probably a lot of that nuclear genetic variation is extinct as well. But you know, that's kind of speculation. I'm not really sure. Let's see, Chris. Okay, that's not a question. Are the tadpoles of different ploidal levels different in size and aggression? Yeah, so I think there's been a couple of studies trying to look at tadpoles and differences. I haven't seen, I don't think anybody's found anything. So as far as ecologically goes, the only thing that's been found that differentiates these two frogs, and it only very loosely, is maybe the height at which they call if they're sympatric. And so that might be a local thing to prevent hybridization. I don't know if it's characteristic of the group as a whole. But as far as tadpoles go, like, I don't think that there's been a whole lot done on it. The tadpoles are, as you probably, oh, sorry, that's for Mike as well. I was thinking of a different Mike. I was thinking of Mike Bernard, who knows a lot about frogs. Might have asked.
I don't know much about them.
Anyway, the tadpoles are really interesting in this group. They've been studied for a long time as studies of phenotypic plasticity, because if you introduce the scent of dragonfly larvae, their tails grow wider, taller essentially, and bright red towards the tips. And how that response differs between diploids and tetraploids, I think would be interesting. I don't think anybody's done that, as well as different population differences as well, because we know that at least in wood frogs, there's lots of local population variation in plastic responses with tadpoles.
All right. Well, yeah, thank you. I had memory of this from some graduate courses I took, and I was curious how that varied. Well, thank you, Will, for the great talk.
Yeah, no, thanks for having me.
Well, we'll go ahead and get on to our next speaker. So Will will close down his screen sharing there and let Shan take over. And if there are any other questions for Will, we'll, as always, have a chance at the end. There'll be an opportunity to ask those then. So just save those questions and keep track of them. And if Will will stick around for a few minutes at the end to answer those, that'd be wonderful. All right. Well, our second talk this morning is from Shengchen Shan. Shan received his PhD from the Soltis Lab at the University of Florida in the spring of 2020. During his PhD, he studied the genetic consequences following whole genome duplication in allopolyploid Tragopogon. He is now a postdoc at the Florida Museum of Natural History working on developing CRISPR and non-genetic model plant systems. And Shan's going to tell us this morning about his work on transcriptome dynamics of the inflorescence and reciprocally formed allopolyploid Tragopogon miscellus and the Asteraceae. And with that, I'll let Shan take it away.
Okay, Mike, can you see the screen and hear me?
Looks good, and I think we can all hear you.
Okay, great. Thanks, Mike, for the invitation, for this great opportunity to present one chapter from my dissertation. It's kind of hard to present after Will because he found that there's only a single polyploid origin in the Treefrog. So I will read out. I will talk more about the multiple origins of a polyploidy in plants. So our work has recently been published on Frontiers. So if you need more detailed information about our work, you can check the paper. And also our scripts has been uploaded on GitHub. So you can check the code through this link. Yeah, so multiple origins of a polyploid. So interesting finding about polyploid research is that many polyploid species are not formed at once, but multiple times. So in a review paper in 1999 from Pam and Doug Soltis, they said the recurrent origins of polyploids are the rule, not the exceptions. So like more than 20 years ago, they already listed over 45 examples of recurrent formations of polyploid taxa that has been reported. So here, just to show you a screenshot of the detailed information of the taxa, the poly levels, as well as the number of origins and the evidence of the multiple origins. So people believe that the multiple origins provide additional genetic diversity in polyploids. First, this is because the genetic diversity of the parents, like shown here, there are 3 different genotypes of diploid A and 2 in diploid B. And also the independent origins of a polyploids, like shown here, can be derived from different combinations of the parents. And also within the polyploids, there is independent assortment and recombinations. And also there's gene flow among different polyploid populations, like shown here. So our study system is Tragopogon from Asteraceae, that is the sunflower family, so which provide a great natural system to study the immediate consequences following polyploidy. So here, like we have 3 diploid species, dubious, perfolius, and pratensis. So the 3 diploid species are native to Europe, where they are largely allopatric. So in the early 1900s, the 3 species were introduced from Europe to the Plus region of North America, where they became sympatric. And then the 2 allotetraploid species formed, that is Mirus and miscellus. So these 2 species are only found in North America, but not in Europe. And based on the herbarium records, people think that they are only about 90 to 100 years ago, 100 years old. So again, Tragopogon provide a great natural system to study the immediate consequences following genome doubling. Okay, so Mirus and miscellus both have multiple regions, like here shown in this map. So the populations are found at Washington and adjacent Idaho. So here, each triangle represents an independent origins of the polyploids. So the diploid parents of Mirus are dubious and perfolius, and the diploid parents of miscellus are dubious and pratensis. So interestingly, as a special case of multiple origins, miscellus can be formed reciprocally and have a distinct inflorescence morphologies. That is, if pratensis is a mom and dubious is a dad, so we end up with a short ligule miscellus. However, if dubious is a dad and pratensis is a mom, so we end up with a long ligule form. So in the field, in most populations of miscellus, we have the short ligule form, like shown here in the orange dots, and only at one population, that is Pullman, Washington, we have the long ligule form. Okay, so previous studies have investigated the genetic consequences following multiple origins, including reciprocal origins in the polyploid Tragopogon. And however, these studies only use a limited number of loci. So 2 years ago, a paper from Lucas Boatwright proposed a new method that they can use the RNA-seq data to access the contribution of different parental gene copies in the expression of the polyploid, and a much larger gene numbers can be analyzed at the same time. So basically, our work is trying to apply these new methods to study the transcriptome dynamics following reciprocal polyploidization in Tragopogon. So our first research questions is how do reciprocal polyploidization events contribute to the genetic diversity in miscellus? And we are trying to analyze the relative expression of duplicate gene copies as well as the total expression of both copies. In addition, we are interested in which genes may be responsible for the different inflorescence morphology between the 2 ligule forms. So here shows a sampling of our studies. So we have both the long ligule miscellus from Pullman, Washington, and the short ligule miscellus from Moscow, Idaho. So as well as their diploid parents from the same locations. So here I have to mention that because pratensis is extinct at Pullman, Washington, so we use pratensis from Moscow, Idaho instead to represent the parental parent of the long ligule miscellus. And there are 3 replicates per population. So here's our method. So we extract the RNAs from the inflorescence and sequence them. So we use Trinity to assemble the reads, and we want to reduce the redundancies from the assemblies. So we use the LACE to generate what we call super transcripts. And we identifying orthologous pairs between the 2 diploid parents using different methods. And we use DSeq2 to analyze the differential gene expressions. And we use the methods from the Boatwright 2018 paper to analyze the homolog specific expression that I will explain in the next slide. And we also annotate those assemblies. So here are the results. So we got lots of transcripts from the diploid parents, And I want to mention that the N50 values is about 1900 base pairs in both diploid species, so which represent a pretty good assembly quality. And also we identified about 12,000 orthologous pairs between the two diploid parents, and we analyzed the differential expression on these 12,000 orthologous pairs. And we then apply kind of a robust filtering process proposed by the Boatwright paper, and we end up with 5,400 orthologous pairs, which are used for the homolog specific expression analysis. Okay, so here are the results. So first, the homologs are duplicate gene copies derived from different parental species in an allopolyploid. So here we have the dubious, the homologs from dubious, and also the homologs from the pertensis. So we want to see the homolog expression patterns in the polyploid, and there are three different patterns. So in this diagram, so the orange bar represents the homologs derived from dubious, and the blue one indicates the homolog from the pertensis. And the short black lines indicate the reads mapped to the homologs, and the abundance for the reads indicate the expression levels. So the first scenario is that there's no expression difference between the two homologs in the polyploid, so which is called there's no bias. The second case is that the homologs from dubious showed a high expression compared to the homologs from the pertensis. So we call this biased homolog expression toward dubious. And the last case is the bias toward the pertensis. So here the result in the two forms. So we find that there's very similar patterns between the short and long ligule forms. So that is the majority of loci, about 80% show no bias. But I want to mention that in both short and long ligule forms, so more loci showed biased expression toward pertensis than dubious. For example, in the short ligule form, 9% loci are biased to dubious, but 11% loci are biased toward pertensis. So these are statistically different. So this is the same in the long ligule form. So we find that we found unbalanced homolog expression bias toward pertensis in both short and long ligule miscellus. So we also try to compare our result with previous studies. So as I mentioned in the Boatwright 2018 paper, so he used the tissue from the short ligule form, but instead of inflorescence that we used in our studies, he used the leaf. So this is the result that about 50% of loci are no biased, and there's the equivalent number of loci showed bias toward either dubious and pertensis homologs. So which means there's no, the homolog expression bias is balanced in the leaf tissue. So this just, we use the same method in the two studies. So this reminds us that the homolog specific expression may be affected by the tissues that you sampled. So then we try to assess the effects of a parental gene expression on homolog specific expression. So there are three different scenarios. First, we call it parental legacy. So that is the expression patterns of the diploid parents are vertically inherited in the polyploid. So this is one case of parental legacy that at one locus, dubious showed a high expression than pertensis, and in the polyploid, we showed the dubious homologs showed a high expression than the homologs from pertensis. So the second scenario is called the absence of a homolog expression bias. That is, at a specific locus, the one parent showed a high expression than the other one. However, in the polyploid, there's no expression bias between the two homologs. And all the other scenarios is called the novel homolog expression bias. And here shows one case that the two diploid parents showed no expression difference, but there is homolog expression bias in the polyploid. So here are the results from the two forms of Tragopogon. So again, the patterns are very similar between the two forms. So that is about 17% of loci showed parental legacies, about 14 to 15 loci showed absence of homolog expression bias, and 15 to 70% loci showed novel homolog expression bias. So Tragopogon is only 80 to 90 years old. So we compared our result with some polyploidy species with a long history. So this kind of analysis has also been analyzed in Gossypia, cotton, and a kind of grass, so which is about 1 million or 2 million years old, which is much older compared to Tragopogon. However, we found that the expression patterns are quite similar. That is, about 60% loci showed parental legacies, and about the remaining 40% loci showed absence of expression bias or novel homolog expression bias. So based on this result, we think that the deviations from the parental patterns arise quickly after polyploid formation. Therefore, the gene expression patterns, which is characteristic of very old lineages of polyploids, can be also found in a very young polyploid system like Tragopogon. Okay, so then we want to compare the homolog specific expression profiles between the two linear forms. So each linear form has 3 different types. There's no bias, bias toward dubious, or bias towards pertensis. So we found that the majority of the loci, more than 80%, showed same homolog specific expression profile between the two forms. For example, there are more than 200 loci showed bias toward dubious in both short and long linear form. But I want to mention that that also means that the remaining, like almost 20% loci showed distinct homolog specific expression, which indicates the transcriptome dynamics following polyploidy. So among those loci, so we are specifically interested in 449 loci, so which we called the lineage-specific biased homolog expression toward the maternal parents. So these loci include, for example, these loci are shown in green, so which, for example, include about 185 loci shown here. So in the long linear form, dubious is the maternal parent. So at these loci, they showed biased expression toward the maternal parents in the long linear form. However, they are not biased in the short linear form. So I'm interested in this kind of loci is because of the cytonuclear incompatibility in the allopolyploids. So that is because in allopolyploid, the nuclear genome here shown in the vertical bars are inherited from the both parents here and here. So however, in plants, in most cases, the cytoplasm is inherited from the maternal parents, like here showing the color. So the maternal parents have a blue mitochondria and a chloroplast. So here in the allopolyploid. So, and we know that the coordination between the cytoplasmic and nuclear genes is very important. So therefore, for example, we have 2 polyploid individuals here, and we are interested in the organelle-targeted nuclear genes. So for example, in the polyploid, these transcripts are from the organelle-targeted nuclear genes. So for example, in polyploid 1, a lot of the transcripts are shown in blue, so which is the same color as the mitochondria and chloroplast, which indicate the same origins. So, and this is showed biased homolog expression toward the maternal parents. And comparatively in polyploid 2, the 2 homologs are not biased. So therefore, because the coordinate expression, so we think, and people think, that the polyploid 1 should have a higher fitness compared to polyploid 2 because the nuclear and the cytoplasmic genes have a more coordinated expression. So we want to see if this is the case in Tragopogon. So we have the 449 loci that showed homolog expression bias toward the maternal parent. And if there is a cytonuclear incompatibility, we would expect that there is biased homolog expression toward the maternal parent among those organelle-targeted genes. And therefore, those organelle-targeted genes should be enriched in these 449 loci. However, here are the results. So we didn't find the proportions of organoid-targeted genes are different between our gene set and the background gene set. So these two numbers are not statistically different. So based on this result, we infer that in Tragopogon, the cytonuclear coordination may not be established immediately following polyploidization, or those organoid-targeted genes showing biased expression toward the maternal parent is still very low. Therefore, it cannot be captured by the enrichment analysis. Okay, so then we try to assess the differential expression patterns in miscellus relative to the diploid parents. So the first expression patterns we call is no change. So here, the orange dot is the D, that is dubious, and the blue one is the pertensis, and the M is the miscellus, the polyploid. So the expression level has been normalized by the transcriptome size. So we found that in both forms, the majority of loci showed no change. The second scenario is called additivity. That is, the expression level of the polyploid is intermediate between the two diploid parents. And here the result in the two forms. And also we have what we call is expression level dominance. That is, for example, here, the expression level dominance of the polyploid, the expression level of the polyploid is equivalent to only one of the diploid parents, but not the other. So there's two different directions. It can be either dominant toward dubious or toward pertensis. The last two scenarios called transgressive expressions. So that is, the expression level in the polyploid can be either can be either lower or higher than both of the diploid parents. So people usually see the non-additive expression include both expression level dominance and transgressive expressions. So, so here in the short legal form, there's a larger proportion of genes show the non-additive expression compared to the long legal forms. So these two proportions are statistically different, so which indicate another evidence of the transcriptome dynamics following reciprocal polyploidization. Okay, so I have 2 slides left. And then we analyze the differential gene expression between the short and long legal forms. So we find that there's only 39 loci showed high expression in the long legal form than the short legal form. And there are 2 loci showed high expression in the short legal form than the long legal form. So we then performed the goal enrichment analysis, and there's only 2 goal terms found among the 39 loci showed high expression in the long legal form. And these 2 goal terms are kind of related. So that is pectin catabolic process and the cell wall modifications. So pectins is a very important component of the primary cell wall, so which can control the cell wall extensibility and therefore might control the cell size and the legal length in Tragopogons. However, this has never been reported in other sunflower family species. So our hypothesis needs to be confirmed. So luckily, we have developed a CRISPR system in Tragopogon. So here, this is the wild type Tragopogon plant. And here, by designing single guide RNAs targeted gene here called is PDS, we got the PDS mutant in Tragopogon, so which is shown the albino phenotype and much shorter compared to the wild type. So this technology could be used in the future studies. Okay, so conclusion. So we found that the overall inflorescence transcriptome profiles are similar in the 2 forms of miscellus, and we also find the dynamic homolog specific expression and total gene expression patterns provide additional genetic diversity. And we found the genes involved in pectin catabolic process may play a role in the different inflorescence morphology of the 2 forms, which can be confirmed using CRISPR later. Okay, so acknowledgement. So I thank Luke Spoderwright for his help in data analysis, and Shengchen, Andrew, and Chaonan for their helps in the lab works, and also the great help from my supervisors, Pam and Doc Sodis. With that, I would like to answer any questions.
All right, well, thank you, Shan, for a wonderful talk. as always, let's go ahead and put those questions in the chat box and ask away.
Okay. Let me see.
They're just giving you praise right now. That's the good stuff. So.
Thank you, guys. You're so nice.
That was a wonderful talk. I have a real quick question that I'll insert in there real quick. So do you see? So you mentioned mitochondria throughout that. So do you see anything chloroplast targeted or specific in these crosses? Maybe you bent all the whole cytoplasmic genome when you're talking, and I just sort of missed that.
So you mean so for the organoid-targeted genes, do you want to distinguish between the chloroplast targeted or mitochondria targeted?
Yeah, I mean, it sounded like originally, I guess maybe when you were just showing those little pictures, it was just the mitochondria, but you looked for both chloroplast and mitochondrial targeted genes. Is that correct in your analysis?
Yes. But none of them are enriched in. So.
And none of them, yeah. It's very interesting.
Yeah, I think I have a question here from Will.
From Will, yep.
Yeah. He asked, did you observe the same or expected phenotypic shift in the synthetic polyploids? Yes, we have the synthetic polyploids, and these show the similar phenotype. Actually, also in the hybrids, the diploid hybrids, they also show the similar morphological difference. Okay. Oh, Doug mentioned that too. So thanks, Doug.
Well, I have another question if no one else does. So as you get ready to CRISPR these guys, I assume that you're going to CRISPR these 41, 39, and 2 genes, right? And knock them out and see what happens. How do you overcome how do you think you'll be able to overcome the ploidal sort of issues with the multiple copies of these genes? Or are you able to have you been able to solve that with your CRISPR techniques that you developed?
Yeah, that's a good question. So so I found that in other studies, although they have different copies, but you can try to design a single RNA target the shared sequence among the different copies. So in that case, you might end up with knock out all the copies at once. But you can definitely do it for multiple times. Yeah, but it's challenging.
And I would guess though that too, maybe because this is an allopolyploid, the copies from each of the parental species may be divergent enough you can generate guide RNAs to target each of those in turn, right, or individually. Is that?
Yes, if you want to do that, you can design a single guide RNA that can target either one homolog but not the other. Yeah, I mean target only one homolog, so that also works. Yeah.
Nice. Well, I really look forward to seeing that work. All right. Well, any other? Oh, I see. And Pam says that you've done this targeting as a test. Awesome. So. Yeah, I think this is awesome stuff, and I really look forward to this. And just so Doug and Pam know, Shan comes to my lab meetings and journal club, and we've been talking about co-opting him to make our brassicas and xanthosmas have some CRISPR power. So hopefully, hopefully we can do that sometime. Anyway, well, I think if there are any other questions for both Shan and Will here, it would be now's the time to ask those. Otherwise, I'll call this one a day and thank everyone for coming. And people can stick around a little bit if you want to chat. I just want to highlight and thank again both Will and Shan for great talks today. It really makes me want to get out there and start digging through some of these polyploid complexes. these are always, this is where I really first fell in love with polyploidy. I think many of us as well, looking at these complex patterns of speciation. It's really fun to see two excellent examples explained by both of you today. And this was of course, our Florida day. So I appreciate both of you coming to us from the Sunshine State today. I do want to highlight our next 2 weeks from today's speakers on October 12th. We'll have a real ecophysiological day with Anthony Baniaga talking to us from Michael Donohue's lab, speaking on ecophysiological differences of coexisting tetraploid and octoploid viburnum, together with Jeanette Witten speaking on the ecological and life history differences between diploids and autotriploids in Townsendia. So two really great talks to look forward to in 2 weeks. And with that, I want to thank everyone again for coming out today, and thank Shannon and Will for two wonderful talks. All right, we'll see everyone in 2 weeks.